So first I'll talk about the human-dog relationship, then I'll move on to talk about oxytocin and bonding, and then I'll just finish up with the specifics of my PhD project. So I wanted to open with the object choice task. So this is a task that has been extensively used in dog cognition studies. So a human indicates the location of a hidden piece of food either to the right or to the left of them, and then it's the dog's task to pick up on these cues and then find the hidden piece of food. So dogs generally outperform both chimpanzees, so humans' closest genetic relative, and wolves, dogs' closest genetic relative, on this task. So it appears that they may have this innate predisposition to interpret social cues from humans, and one of the reasons being is because they gaze more at humans to solve problems than do other species. So what this task demonstrates is that there is this special relationship that exists between dogs and humans that's absent in chimpanzees, which are closer related to humans, and obviously has formed through the process of domestication because it's absent in the ancestor of the dog, the wolf. And there are many benefits to this relationship. So in 2008, Horowitz published a review on some of the health benefits, including reduced risk of cardiovascular disease, reduced anxiety, reduced post-operative pain, increased sociality, etc. And in the year 2000, Hetty published a longitudinal study of pet ownership and doctor visits, and found that medium to long-term pet owners had 10% less doctor visits than new owners or people that didn't own pets at all, which equated to 3.86 billion Australian dollars saved in national health expenditure in a one-year period. So despite these benefits, it is the sad reality that many dogs end up in shelters. So in 2004, Marston published a study revealing that there were over 20,000 admissions just to 3 Victorian shelters in a one-year period. 83% were stray dogs, and 15% had been relinquished by their owners. In Victoria, there's a mandatory 8-day holding period for all stray dogs. After this period, or immediately for relinquished dogs, they're temperament tested for suitability of adoption. If they're considered unsuitable, they're immediately euthanased. And if they're considered suitable, they're held for another 28 days, in which time they can be adopted out before euthanasia. So 31.5% of dogs entering a shelter for the first time are euthanased, but this number jumps up to 40% for dogs that are sold and then subsequently returned to the shelter, so dogs entering for the second time. And 22% of these returns are due to behavioural problems. And there is a direct relationship between undesirable dog behaviours and poor quality human-dog bonding. So then what goes wrong? As McGreevy and Bennett highlight in their 2010 paper, dogs are continually being bred as companion animals, despite lack of owner feedback on the success of the dog as a companion animal, and also breeder accountability if the dog is unsuccessful. Which begs the question, what are people looking for? So King et al. In 2009 published a survey of close to 900 participants on what their ideal dog would be. 5 behavioural subscales were created, so calm, compliant, sociable, healthy, energetic, faithful, protective, socially acceptable, and non-aggressive. And the sociable, healthy subscale was considered most important, with a mean score of 4.36 out of a possible 5. So items included: my ideal dog shows affection towards me, enjoys being cuddled, enjoys being petted, friendly with other dogs, safe with children. And it was considered either important or extremely important for the ideal dog to be affectionate for 92.7% of participants surveyed. So what this is potentially indicating is that what people are looking for are dogs with high oxytocin function. So what is oxytocin? Oxytocin is a hormone in the body, so it's involved in reproductive function, stress response, and immune function. And it's also a neuropeptide in the brain, so involved in emotional regulation, mood, and bonding. And it's been found that oxytocin in the periphery of both humans and dogs increases when humans and dogs interact with each other. So the first graph here is from Hanlon et al. In 2011, and it shows that just after 3 minutes interacting with their owners, the dogs plasma levels of oxytocin are significantly increased. The owner's level at 3 minutes was not significantly different to baseline, but the peak level, which occurred at either 1, 3 or 5 minutes, was significantly different to baseline. However, in 2009, Nagasawa showed that there are differences in oxytocin release. So this was urinary oxytocin, but it showed that owners with greater rated satisfaction of their ownership of their dog had more, significantly more oxytocin in the urine following a 30-minute interaction, and their dogs gazed at them for longer durations during that 30-minute interaction, whether the owners were looking back at them or not. So this once again implicates gaze as an important factor in the human-dog relationship, as we saw before in the object choice task. But I should mention here that it's not always a good way to measure peripheral levels of oxytocin as a measure of central oxytocin function, which is involved in bonding. And the main reason is because oxytocin does not readily pass the blood-brain barrier and can be independently released from tissues in the periphery and also in the brain. So one way around this problem is to actually administer oxytocin intranasally and then modulate the system in that way and then evaluate things like behaviour. And this has been the method used in many human studies. So in 2010, McDonald and McDonald published a review of some of the effects of intranasal oxytocin. So decreased cortisol, reduced amygdala and fear circuitry activity, decreased trust, decreased anxiety, increased trust and generosity, increased emotion detection and increased gaze towards the eye region. However, just last year, Bartz et al. Published a review of some of the more social cognition and prosociality studies and found that just half of the studies had a significant effect. And while the majority increased prosocial behaviours, a sizable minority, 43%, actually decreased prosocial behaviours, which led the authors to the social salience hypothesis, which basically states that oxytocin increases the salience of social cues. So in studies involving significant others and in groups, you see an increase in prosociality. But in studies involving deceit, competition and outgroups, you might see a decrease in prosociality. So the conclusion from their paper was that context and person matter as to the effect of oxytocin. So some of the personal factors that influence oxytocin function are past experience. So another study that involved owners coming home from work and interacting with their dogs and then having plasma levels of oxytocin taken demonstrated that 2 participants had levels that were 2 standard deviations greater than the mean, and they were the only 2 participants with the most experience of bonding, so one with small children and one with pets. Stress. So oxytocin is known to decrease stress and stress hormone release, but is also released in response to stress, which can also make measuring oxytocin somewhat confusing, particularly in the absence of any measure of stress, because you don't know whether the oxytocin level is high because the individual is stressed or because they're quite relaxed. Sex hormones, so rodent studies have shown that pups that receive high amounts of maternal attention have a propensity to have more oxytocin cells in their brain, but more so in females than in males. And adults that receive central oxytocin administration facilitates pair bonding, but more so in females than in males. And also the oxytocin receptor gene. So in humans, a variant of this gene has been associated with autism, decreased sensitivity of mothers to their toddlers, decreased physiological response to infant crying, and decreased sociality. It's currently unknown whether there are any variations within the canine oxytocin receptor gene. So this is going to be one of the objectives of my PhD, to look at microsatellites close by to that gene to see whether an association with behaviour can be found. Okay. So my PhD is broken up into 2 parts. So just for the first part, we're going to be testing 80 animals housed at a shelter in Victoria. The day before the experiment, baseline levels of stress will be taken, so heart rate. They'll then be taken to the testing room for 10 minutes, also to familiarise the dog with that room. And then they're going to have saliva samples taken for subsequent cortisol analysis, and a blood sample for the neutrophil lymphocyte ratio, another stress indicator, and then also a sample of blood for the genetic analysis. And then on day 1, they'll be fitted with the heart rate monitor, taken back to that same room where the student researcher, myself, will already be, and the dog can approach for up to 10 minutes of calm positive interaction, which will be video recorded for later behavioural analysis. The dog will then return to its compound, have blood and saliva samples taken again. So day 2 will be repeated that same procedure, except for dogs in group 1, they'll receive oxytocin intranasally 45 minutes before that interaction, or for dogs in group 2, saline control. Day 3 will be repeated the procedure on day 1, so no drug intervention. And day 4 will be repeated the procedure used on day 2 with the groups swapped, so each dog will get a treatment, the same treatment. So what we're expecting to see is that on the day the dog receives the oxytocin, there'll be more bonding behaviours, less stress, and also that their bonding behaviours will be related to variations in the gene, should we find any. So hopefully all these dogs will be adopted out, allowing for part 2 of the project. So on the day that they get adopted, owners will be asked to participate in the study. And then approximately 1 month later, they'll complete an over-the-phone questionnaire containing the Monash Dog Ownership Relationship Scale, Pet Attachment Questionnaire and other information about pet ownership, so that we can gauge the strength of the new bond and see whether we could have predicted the strength of the bond from any of the factors inside the shelter. So the implications from part 1 are to improve our understanding of the neurobiology of human-dog bonding. May also be possible to improve shelter animal welfare, so for example stress reduction by the administration of intranasal oxytocin. And it may be possible to selectively breed dogs with the appropriate genetic constitution to improve dog-owner relationships if we find that there is a genetic link there. So part 2, there's a potential to improve methods to match dogs with their new owners, so according to owner needs and canine factors inside the shelter. And also it may be possible to provide a novel method to enhance the human-dog bond post-adoption by the administration of intranasal oxytocin, so thereby reducing return rates to the shelter and thereby reducing rates of euthanasia. And lastly, I'd like to acknowledge other people involved in the project, my supervisors and the staff at Animal Aid. Thanks, that's it.